Steve Green is going to talk to us about the hidden hearing loss, synaptopathy, and tinnitus.
Steve, thanks. I'm not sure I need this, but I'll put it on. I lecture in the undergraduate neuroscience course, so I've tried to develop a louder style. Anyway, so Rich asked me to kind of do an overview of some issues in noise damage and possible consequences to tinnitus. And so this will be kind of an overview, and if people want a little bit more information about some of the topics, we can talk about it in the Q&A or in the break. And I'll talk mostly about work in a number of labs, a little bit about some work in my lab here at Iowa. Okay, so the key word here is synaptopathy, and just as which is a little bit of explanation, I think most people understand that noise damage can impair hearing by causing the destruction of hair cells, the sensory cells in the inner ear. And in my picture from my lab, these would be the hair cells that are here in blue, and they're contacted by nerve fibers here in red that then carry the information into the brain. Anyway, this has been a type of noise damage that's been explored for years, and it's known that it can be a trigger for tinnitus. And this is just an example, electron micrograph of a dead outer hair cell. Now, synaptopathy, or a noise destruction of synapses, is a more recently discovered phenomenon. It's really only been described in great detail by Charlie Lieberman and Sharon Kajawa at Mass Eye and Ear, really just in the last 10 years or so. The phenomenon was originally discovered by Rémy Pujol and Jean-Luc Puel in Montpellier. And the idea seems to be that very loud noise, or loud noise, causes an increased activity at this synapse between the hair cell, between the sensory cell, and the nerve fiber that's going out into the brain. And here you can see in the electron micrograph these little synaptic contacts between the hair cell and the nerve fibers that contact it. So as a consequence of noise, you get excessive release of the neurotransmitter. And for the synapse aficionados, you know that even in the brain, excessive release of this particular neurotransmitter can have devastating effects, can be highly neurotoxic, and that can happen, for example, during stroke or in some neurodegenerative disease. In the cochlea, the consequence is to cause entry of calcium ion into these little terminals, and that results actually in the destruction of the terminals. As Pujol and Puel first noted here in electron micrograph, you can see that these things just become swollen and vacuolated and degenerate. And this paradigm that we use in my lab, this is actually data from my lab, but the paradigm originally developed by Charlie Lieberman and Sharon Kajawa. What we've actually done is count individual synapses. So each one of these pairs of red and green dots is a synapse between the sensory cell and the nerve fibers. And after this moderate noise exposure, which you can see a reduction in the number of synapses at these various locations in the cochlea. These experiments are done with mice. The hair cells survive. So the noise in this case is not loud enough to cause any destruction of hair cells, although the synapses degenerate, but we can actually go through and count all of the outer hair cells, the inner hair cells, and you can just see that there's still a nice line of hair cells here. None of them have died. What is the physiological consequence of this? If we look again, the paradigm originally developed by Charlie and Sharon, but this, again, data from our lab. So noise exposure is day 0. We've measured the auditory brainstem response, the ABR, and what we're plotting here is the ABR wave threshold, looking in particular at wave 1. So we've looked at the threshold. This is the pre-noise exposure threshold. This is the threshold just a day or two after noise, and we can see that there's been a threshold shift of about 40 dB and generally about 35 to 40 dB with this particular noise exposure. But if we wait another couple of weeks, we find that there is no permanent threshold shift. The threshold has returned to the pre-noise level, which is consistent with the fact that the hair cells themselves are intact. Nevertheless, there is a hearing impairment, because if we look at the amplitude of wave 1 of the ABR is actually derived from the activation of the nerve fibers in the cochlea. So if we look at the amplitude of wave 1 as a function of stimulus level, and so we're looking here at the growth of wave 1, we can see that the growth is severely reduced after the noise exposure. And this seems to be correlated with the loss of synaptic contacts. So we have an unusual hearing impairment here, or unusual up to this time, where the ABR threshold is in fact not affected, but wave 1 amplitude growth is reduced. And we now can think about whether this might be related to tinnitus. I should say that this synaptopathy has been explored just really almost entirely in mice, a few experiments on guinea pigs. Does it occur in humans? There's still very, very little data on that. This data is from a paper published last year in which some postmortem studies were done on with human material, counting the number of synaptic contacts. For technical reasons, this is really very difficult to do, and they've only got 4 subjects here. But what they did note in this study is that the number of nerve fibers and the number of synaptic contacts seems to decrease with age. So here we have a 54-year-old, a 67-year-old, and a 89-year-old. Individuals, and we can see that the number of synapses is actually decreasing with age, although that was for these 3 individuals, 2 cochlea from this one person. We can see here's another individual here that didn't seem to fit the pattern. But the speculation is that this synaptopathy described in animal studies also occurs in humans. And these are just figures of the cochlea from these particular locations. Now, is there a non-invasive way of determining whether synaptopathy occurs in humans? And again, a very recent study suggests that it may be possible. So wave 1 amplitudes, or the growth of the wave wave 1 with increasing sound levels, is very, very easy to measure in mice. It is actually an extremely easy measure, very difficult, as many of you know, in humans, and so not the kind of thing that you want to have to depend on. However, it is possible that a much more robust measure, which is wave 5 of the auditory brainstem response, which increases in its latency with increasing noise levels, that actually may be a possible clinical measure of synaptopathy. What's shown in this paper, at least, is that it is correlated with the decline in growth of wave 1. So this would be a decline in the growth of wave 1 in microvolts per decibel, and this would be increasing synaptopathy. And you can see this correlation here with 10 human subjects, 10 volunteers, where the latency change in milliseconds per dB, the latency change as the noise level is increased, also declines with the declining growth rate. And if people want the explanation for that, we can talk about it in the Q&A, but it has to do with a preferential loss of low spontaneous rate fibers as a consequence of synaptopathy. Now, now, for tinnitus, and all of you know this, you've probably heard a much better explanation of this from Dr. Melcher. One of the favorite ideas for the cause of tinnitus is that as you lose hearing, as there is a hearing loss, which is usually preferentially at higher frequencies, there is an increased gain. You basically turn up the gain. And so that results in a mean activity level that is normal, however, at the expense of an increased spontaneous rate of activity in the central nervous system. And it's believed that tinnitus is due in large part to elevated CNS spontaneous activity. Now, clearly, if you've lost hair cells, there's going to be reduced input into the CNS, and therefore there would be an elevation of gain to bring the mean level of activity up to the normal level. But you might imagine the same thing would certainly be true in synaptopathy. We've seen a much reduced amplitude in activity in the auditory nerve. That should also translate into reduced input into the central nervous system, and consequently you would also have to ramp up the gain and increase spontaneous activity. So it's certainly possible that tinnitus could be a consequence of synaptopathy, not just of kind of the conventional famous hearing loss that involves loss of hair cells and increase in auditory threshold. So the idea, now how do you measure that? One strategy that Shetty and McAlpine took in a paper published about 5 years ago is to figure, well, fewer synapses is reduced ABR wave 1 amplitude. That certainly checks. We've seen that ourselves, and it's been extensively published by others. But if there's increased spontaneous activity, there might be an increase in activity in the amplitude of wave 5, which is a robust measure in humans. And in looking at a number of subjects, they in fact found that this was the case. So looking here at a control group and at a tinnitus group, they reported that looking here at comparing the mean tinnitus spectrum as a function of frequency with threshold, you can see that there is not a good correlation, not necessarily a good correlation between tinnitus and an elevated threshold. However, there did seem to be a correlation between tinnitus and a reduced wave 1 to wave 5 ratio, which again would be consistent with the idea of synaptopathy, although again without a non-invasive measure of synaptopathy in humans, which is, I've just given you a couple of strategies by which that might be determined, but there's nothing definitive yet, so we don't know for sure, but certainly this is very, very suggestive that tinnitus could be a consequence of synaptopathy, which remember is occurring at noise levels much lower than those that are associated with threshold elevation, and may even be below levels that are regulated by OSHA or below the NIOSH recommendations. Okay, so can anything be done about it? I'll close briefly with a couple of attempts that we have now in trying to clinically treat or prevent synaptopathy. So to recall, synaptopathy can be caused by sound levels that don't damage hair cells and that don't raise ABR. And of course, we have the issue that ABR wave 1 amplitude is difficult to measure in human subjects. So in terms of now trying to tackle this clinically, and really we're at the level still of basic research, we're asking the questions, how does noise actually damage the synapses? Can the synapses regenerate? and can the damage be prevented? And if we have an understanding of how the noise damages synapses, maybe we can figure out some strategies for prevention. So my lab looked at this question some years ago and found, using initially an in vitro model, that in fact synapses regenerate very, very poorly after they're damaged. And this is consistent with a number of in vivo studies. But what we did find is that addition of a neurotrophic factor, so a survival factor, that tends to be produced in the hair cells, and that a number of labs, including ours, have shown can promote survival and prevent degeneration of cochlear neurons, that addition of the neurotrophic factor actually increased the number of synapses that could regenerate to up to about half or more of the original number. Recently, Gabriel Corfus's lab did an experiment where they exposed mice to noise and injected this particular neurotrophic factor, NT3, transtympanically onto the round window, so that presumably in at least some of these mice, it got into the cochlea, diffusing through the round window. And what they did find, at least in some of the animals, was an increase in the number of synapses, or presumably in triggering synapse regeneration. So if we look here at the 32 kilohertz location in these mice, this would be the normal number of synapses. This is the number of synapses after noise exposure. But in at least a subset, what they called the effective subset of the mice treated with NT3, they saw a greatly increased number of synapses, evidence of synaptic regulation, presumably triggered by the NT3, although there was a subset of mice that showed actually very poor synapse regeneration. What the difference is among these groups, we don't know, but again, this is an extremely recent paper, 2016. And they also showed corresponding improvement in the ABR wave 1 growth, here again at 32 kilohertz. In terms of trying to use what we know about the mechanism by which the synapse is degenerated, my lab has started looking at methods that might be used to prevent the damage to begin with. And of course, the strategy was to try to prevent calcium entry into the synapse. It's believed that that is the ion that is triggering the damage, that's triggering the degeneration of the synapse. So in my lab, we've looked carefully at the routes by which calcium can enter into the synapse. And for the synapse aficionados, I've got all the background here. But to get to the punchline, what we found is that entry through one particular channel seems to be responsible for the damage caused by noise and by excessive activity at the synapse, by excessive release of neurotransmitter from the hair cell. That would be the case with noise. So we looked at inhibitors or blockers of this particular channel, and we actually found, and I'm skipping over a lot of experiments that we did in culture to develop the strategy. These experiments that were actually done in mice, into which we infused the blocker intracochlearly, so we actually put a cannula into the mouse cochlea and infused the drug. What we found was, in fact, no loss at all, no reduction at all in ABR wave 1 amplitude growth relative to these mice that received only vehicle and without the drug. And if we count synapses, so some of this data you saw in an earlier slide, the reduced number of synapses after noise exposure in mice, we can see here that that vehicle alone had no significant effect, but infusion of the drug actually entirely prevented the loss of synapses. So at least studying the mechanism has given us an idea of strategies that might be used to prevent the damage. So in summary then, with regard to regeneration, NT3 seems to be necessary for synapse regeneration on hair cells, and application of NT3 can promote regeneration. And in terms of prevention, selective blockade of the appropriate channel can be a possible approach for prevention. But keep in mind yet that these are all still very preliminary studies, and the safest way to avoid damage is to keep the volume low, especially when you're using these things, which put out noise that is really sufficient to cause synaptopathy. Okay, so I'll terminate. I don't know if we have time for questions.
Short questions?